thank you very much for inviting me to talk about a different aspect of sex and sexual reproduction that I find very interesting. This work is kind of based on one of my heroes' work, which is Barbara McClintock, who discovered a long time ago that it turns out that the genome, which was once thought to be fully static genome in which genes didn't move, there's a different type of gene, which we now know is to be a transposable element or a jumping gene, that actually does move. And her work came out of this work here, where you can actually see in these corn kernels that there's this variegation in this pigmentation, and this variegation in pigmentation occurs when you have, for example, a pigmented gene sometimes has another type of gene, also known as a transposable element, inserted into it, and that leads to loss of pigmentation. But every once in a while, that gene can jump out of that pigmentation gene and lead to restoration of this pigmentation. And so Barbara McClintock was the one who actually discovered this particular class of gene called a transposable element. Now, transposable elements can be considered selfish DNA because, and they have a very unique capacity to actually move within genomes. And I won't talk about the exact mechanisms by which they do this, but the most, in my mind, one of the most important things to understand about transposable elements is they're actually exploiting sex itself to move. All right? And so in 1982, Donald Hickey, at this point, people sort of thought of these jumping genes as sort of not necessarily harmful. They just can maybe move around a lot in the genome without harm. But Donald Hickey in 1982 showed that transposable elements actually can increase in copy number even if they're harmful within populations. And that's because of the way that they can move in sexually reproducing organisms. And the specific thing here is during fertilization, genomes, which may be unrelated to each other, at least within respect to close ancestry, genomes come into the contact of other genomes. And it's the capacity for selfish DNA to move from one genome to another, or specifically colonize new genomes, that allows transposable elements to spread within species and within populations. And they can actually spread even if they're harmful. So they can have a net negative effect, but they'll increase in copy number as long as there's this rounds of outcrossing. And so you can kind of consider, as I said, transposable elements are basically genes that can copy themselves. And so you can imagine that one may invade a particular species or a population, and you'll end up seeing this sort of replicative process, and it can actually quite quickly fill up a genome. Another thing to know is that transposable element insertions can actually be harmful. So as we're doing more and more genome sequencing, it's been identified there are at least multiple cases of colon cancer that have been identified in humans that are caused when a transposable element known as the line 1 element has been shown to insert into genes that are involved in regulating the cell cycle, and this can eventually lead to colon cancer. Now, one of the things we're trying to understand in my lab, which from a broader sort of theoretical perspective, is based on these strange observations that some genomes are completely filled with transposable elements. And so, the black salamander is probably about 50% TE. Corn is about 60% transposable element. And one of the jokes that a friend of mine often says is that you can consider corn to be more transposable element than corn. And then here's Barbara McClintock winning the Nobel Prize, and humans actually probably have more than 60% of their genomes comprised of transposable elements. You can contrast that to other species such as pufferfish or fruit flies, which is what we work on mostly in my lab, which have much lower transposable element content. And it's not super well understood. There are some ideas as to why some species have low transposable element content and other species like us have such high transposable element content. Some insight can be obtained by doing what you can consider genome paleontology. So the thing that's interesting about transposable elements is as they move through a genome, they leave a record of their invasion. And so here's a sort of a recent event which may have occurred within a population, which a large proliferation of transposable elements occurred maybe perhaps recently. But after this invasion event and proliferation event, these transposable elements will then begin to accumulate mutations. So there's a mutation there, and there might be some more mutations. And over time, these transposable element copies, which are originally identical to each other begin to diverge in sequence from one another. And what that means is you can actually trace the activity of transposable elements in genomes by looking at genome sequences and actually analyzing the divergence among copies to look at patterns of invasion and loss over time. And what's really kind of one of the most interesting things that came out of the human genome sequencing project in the early 2000s, and also in contrast to the mouse, is, for example, there are these different types of transposon families that you can see, and this is back in distant time, and this is in recent time, and this is sort of the abundance of these different age classes of these different transposable element families. And going in distant time, there's sort of this pattern activation, and there's an expansion of one particular class of transposable element. And actually, in very recent time, there seems to have been a crash in which the transposable element activity in human lineages seems to have subsided. Now, if you contrast this to mouse, actually there's sort of been this ongoing pattern of transposition that seems to be ongoing to this day. And so these patterns of proliferation are I think, quite interesting. Now, because transposable elements can be harmful, what has become apparent in the past about decade is that there are actually mechanisms in place to protect the genome from transposable elements. And one of those systems is a system based on small RNAs. And I'll talk more about these small RNAs in a moment. But the major class of small RNAs are piRNAs, which are called, or known as Peewee interacting RNAs. So just since this is a public lecture, just want to make sure, get everyone on the same page. As we know that DNA is the source of RNA, and then RNA is the source of protein. But it's actually these RNA molecules, because they have the capacity to, through base pairing, they're almost used as these sort of targeted antibodies that target transposon mRNAs for a degradation reaction known as a slicing reaction. So there's a particular class of RNAs which are small and antisense, they're about 23 to 30 nucleotides long, and they form a complex with a bunch of different proteins called Peewee proteins. And those can then recognize transposable mRNAs as dangerous passengers inside the genome and target those transposon mRNAs for something called slicing, which actually can lead to the degradation. Now, how is this particular class of small RNAs, known as Peewee interacting RNAs, generated? All right, because it's obviously the case that you wouldn't want to be performing this reaction against normal genes. You want to actually identify and target transposable elements specifically for degradation. And in some ways, this actually does really have a lot of analogies to the immune system, in which you have to identify an invader and then target it. All right, so it is really a genome immune system. And the way that these piRNAs are generated is through basically the evolution, it's amazing, has identified the Achilles heel of transposable elements. And the Achilles heel of transposable elements is they move, as Barbara McClintock found a long time ago. Normal genes don't bebop around the genome, but transposable elements do. And so there, you can imagine in a genome there would be a proliferation event, and this particular transposon is copying itself here and copying itself here. But there are particular regions of the genome known as piRNA clusters that specifically recognize genes that insert into them, all right? And then instead of allowing that particular transposable element to generate a normal mRNA, which would then make a protein, which would then allow it to copy itself, actually then that particular transposon will end up producing a class of antisense, or the reverse complement sequence, that are about 23 to 30 nucleotides, and that can lead to the repression of transposable elements. Actually, not only is this repression mediated by that slicing reaction that I talked about, is in probably a more important, perhaps, mechanism of repression, is that this transposable element piRNAs, in complex with those Peewee proteins, enter the nucleus and target multiple diverse copies of the same family for transcriptional repressing, meaning they don't even allow these mRNAs to be expressed. And you can almost consider this sort of a strategy of fratricide, in which one member of a family is used to rat out the rest of the family and actually use that to actually target that for degradation. One thing that I find interesting, and this is work I'll talk a little bit about, is that it turns out, at least in fruit flies, it seems like it's probably a little different in mammals, in fact, but at least in fruit flies, because these piRNAs that target transposable elements are RNA and are loaded into the egg. Okay, so here's, for example, an egg, and it's being fertilized by this particular sperm. And this particular sperm carries a particular transposon family that the mother also has. And so this is the mother's genome right here. And there's a transposable element that is the source of piRNA. And so that's what this little hexagon and wavy line indicates. All right, so what we have here is that the mother, which has a particular transposon family, generates these piRNAs that target for repression transposable elements that will also be inherited from the dad. But you can have a situation in which the mother may not have been exposed to a particular transposable element family. And because she hasn't been exposed to that particular transposable element family, she will not be the source of piRNAs. And so what you end up having is a situation in which transposable elements inside the genome of dad find themselves in this sort of de-repressed environment. And this can actually induce what we can call a hybrid dysgenesis or hybrid sterility. And so in this particular picture here, this is a male, this is Drosophila virilis that has a normal testes. They're pigmented so you can see them nicely through the wall of the abdomen, and there's where the germline stem cells are. There's a highly degenerated testes here and this is arising from the hyperactivation of transposable elements that proliferate in the germline, and the exact mechanisms for which the sterility is induced is not very well understood, but we think it is basically a programmed cell death apoptotic response that occurs in the presence of DNA damage. If you compare different flies that have different transposon abundances, what did I just do? Try this. Turn something off. There we go. Okay, if you compare flies that have different assemblages of transposable elements and look at the pools of piRNAs that they load, in this particular case, this is arising when there's a large number of transposable elements that are inherited from dad that are absent in mom. And so you can actually do small RNA sequencing of these eggs, and you can actually look at the piRNAs that are loaded differently between moms and dads. And so for this particular assemblage, for this particular syndrome of hybrid dysgenesis, which I just discussed, there's what I'm showing on this diagonal here. This is actually the measure of those piRNAs derived from different transposable element families from one strain of flies, of Drosophila virilis, and then measured relative to the abundance of piRNAs coming from different transposable elements from a different strain of flies. And this is when there's this asymmetry between the piRNA pools that are loaded from mom and the transposons loaded from dad, this releases this germline sterility. So what you can see here is in red are all the transposable elements that are abundant in a strain of fruit flies in which males have this capacity to induce this sterility event. And so you can see that with this large assemblage of red transposable element families that are abundant in the inducer strain, there's this corresponding asymmetry in the piRNAs that are loaded between those two strains, between mom and dad. This form of genome defense, which is transmitted across generations, has an interesting side effect, all right? And this is what we, in our lab, we call genomic autoimmunity, because not only are these transposable elements regulated by these piRNAs, but sometimes this machinery of genome defense has an off-target effect. And what I'm showing here is in this particular case, there's this red transposable element that's being targeted by these piRNAs. But every once in a while, genes, normal genes, get caught in the crossfire, okay? So when transposons land near genes, there are interesting mechanisms that are not super well understood, but when a transposable element can land near a gene, not necessarily turning the gene off through insertion, it can actually lead to the induction of generating piRNAs that target that gene itself. So the gene here is in blue, and piRNAs can be generated to target the gene as well. And this is interestingly evident in this syndrome of hybrid dysgenesis, in which we've been characterizing differences in the piRNA profile with respect to TEs. There are also interesting cases that have been observed in which there are differences between strains in the ways in which other genes, normal genes, are targeted by RNAs. And there's this one gene called center divider. In one of the strains, you can see this is actually a mapping of these RNAs, and this measures the abundance of both sense and antisense RNAs that target a particular gene known as center divider. Now, in one of these two strains, there's actually an abundance of RNAs that are not just targeting transposons, but targeting the gene itself. And so this is a measure of both sense and antisense RNAs that target the center divider gene in this strain, but not this strain. And one thing you can also do is from transcriptome sequencing is you can look at what is the corresponding level of expression for the gene that seems to be the target for RNAs. And so this is what this measure is a measure called RPKM. And you can see that when there's no RNAs targeting this particular gene center divider, it's at pretty good levels of expression, 78 RPKM. But when RNAs are targeting this gene center divider, it's lowly expressed. Now, I pointed out that there's an asymmetry in which the ways that moms and dads provision RNAs to the next generation. And so if you actually look at the gene expression of genetically identical females that are occurring from reciprocal crosses, when you take mothers that have this RNA profile and you cross them with dads from this strain, and you look at gene expression and RNA abundance, you see something interesting. This is the daughter of this mother. These two are daughters actually of this mom. This is a daughter of this mom. This is a daughter of this mom. But these two females are genetically identical because they're from reciprocal crosses, okay? And what you see is that when mom is not targeting the center divider gene and is not generating RNAs for center divider, this pattern of expression is maintained. There's a high level of expression, and you basically don't see any RNAs targeting center divider. But if you look at the daughters of this mom, all right, mom here is targeting center divider, making RNAs that turn it off. If you look at her daughters, you can actually see that this pattern of RNA targeting, this off-target effect, is maintained in the next generation. So even though these are genetically identical females, this center divider gene is turned off, while this center divider gene is turned on. So one thing that we're generally interested in my lab is to understand the sort of significance of this really important tension, right? There's this tension between genome defense, but there's also a cost of genome defense, which is this form of genomic autoimmunity. And how this balance is maintained in populations is something that's of great interest to us. In addition, since this is a center for evolutionary medicine, I'd like to sort of broaden this out a little bit and ask a sort of a broader question about the nature of this conflict within the germline. A lot of times we sort of acknowledge that it's difficult for couples to get pregnant. So, it's probably about 10% of couples who are trying to get pregnant have an extreme difficulty in this. And from an evolutionary perspective, that's really remarkable. I mean, I sometimes forget how remarkable that is because, really there's only two things you need to do from a sort of a Darwinian perspective, right? Is like eat and maintain yourself and reproduce. We're all really good at eating and maintaining ourselves, but this is a very low level of fertility if you think about it. And so one thing I sometimes wonder about is to what degree this problem could be explained, at least in part, by these interesting dynamics of selfish elements that are actually persisting within the genomes. And RNAs are also observed in mammalian systems, not just fruit flies. And so you can actually have genetic knockouts for machinery that generates RNAs. And so they have the names of the machinery in mice, Millie and MeWe2 and MeWe. All of these knockouts result in male sterility. And so it's interesting to think about what's actually going on. We don't really understand the genetic basis for why these male mice will be sterile. It's interesting also that these actually lead to no observable phenotype in females. So it's a broader question I would like to sort of open up. And so with that, I'd like to thank you for inviting me to give this talk. And I would like to also introduce Lucas Hemmer, who's sitting back there. And he's going to be presenting a poster, or he has, and I'll be talking further about this work, about understanding the recombination landscape in the face of this hybridogenic phenomenon. With that, I'll take questions. Yes, Jamie.
Is there any possible connection between sort of the male infidelity that happens in this medical result from these pirating and Haldane's rule? I mean, are we overlooking something?
Well,
Question.
could this play a role in Haldane's rule? I think probably in this particular case, this would be an example. Well, so people, at least in fly world, have tried to identify the genetic basis of Haldane's rule, and it doesn't seem to be the Y chromosome, okay? And so, you know, whether the Y chromosome, because the point is that male heterogametes, is the Y chromosome replete with transposons, and therefore do we have this particular fertility defect in those male mice? Is it because of the Y chromosome? I guess that's not fully well understood, but I think probably the source of Haldane's rule is not the Y, but is probably some other kind of more complicated interaction between X and Y in autosomes.